On June 5, 1991, Space Shuttle Columbia lifted off from Cape Canaveral, carrying a payload that seemed almost absurdly small. Inside a sealed temperature-controlled chamber were 12 moon jellyfish, each about 2 inches in diameter, drifting in a bag of seawater. This was mission STS-40, Space Lab Life Sciences 1, and the jellyfish were there to answer a question that had quietly bothered biologists for decades. What happens to a creature that has never known anything but Earth's gravity when you suddenly take gravity away? The experiment was called JELLY. Its principal investigator was Dr. John R. H. Baker from the University of Texas at Austin, a biologist who had spent years studying how simple nervous systems process orientation. The jellyfish species was Aurelia aurita, the common moon jelly, an animal so transparent you can see its entire digestive system through its bell. It has no brain, no centralized nervous system to speak of, yet it navigates the ocean with a precision that engineers still envy. What Dr. Baker wanted to know was whether that precision depended on gravity at a cellular level, or whether it was merely a learned behavior that could adapt to any environment. The textbook answer would suggest adaptation. Life finds a way. Organisms adjust. But here is what the textbooks ignore. When the chamber was sealed and the shuttle cleared the atmosphere, something unexpected started happening inside that little plastic container. Picture a jellyfish swimming. Its bell contracts in a smooth, rhythmic pulse, pushing water out behind it and propelling the body forward. On Earth, that pulse is tuned by tiny calcium crystals inside the jellyfish's statocysts, gravity sensors that tell the animal which way is up. In orbit, without gravity, those crystals floating inside fluid sacs can no longer settle. They drift. And without directional information from the statocysts, the jellyfish cannot tell which end is the surface and which end is the abyss. What Dr. Baker observed after the 12-day orbital period was documented in the Journal of Experimental Biology in 1992. 9 of the 12 jellyfish had developed disoriented swimming patterns. Their bell contraction amplitude dropped by 31%, from an average of 6.1 millimeters on Earth to just 4.2 millimeters in orbit. Their pulse frequency fell from 0.78 Hz to 0.42 Hz, and when they did attempt to move, there was a measurable delay of nearly 1 full second before they initiated any swimming motion at all. A second of hesitation might not sound like much, but for a creature whose entire survival strategy depends on rapid escape from predators, it is an eternity. That 1 second of hesitation is exactly the kind of detail the textbooks bury in footnotes. But if you felt that pause, if you recognized that something is being hidden from the clean story you were told, then you are exactly the viewer this channel exists for. Subscribe if you want to keep uncovering experiments like this one, the ones that quietly shatter the textbooks you thought were settled. And drop the word eternity in the comments, because that 1 second changes everything about how we read biological failure. The findings carried a deeper implication. If a creature as simple as a jellyfish could be disoriented by the loss of gravity, then the entire cascade of biological systems that depend on gravity sense might be more fragile than anyone had assumed. But this is where the story pivots. Because the jellyfish did not just reveal a biological vulnerability, it revealed a solution. Dr. Baker's team published a follow-up histological analysis in 1995, co-authored with Miller and Satterfield in the journal Life Sciences. They dissected the returning jellyfish and compared their statocysts to ground controls. What they found was a 42% loss of otolith mass in the space-flown specimens. The gravity-sensing crystals had literally atrophied after only 12 days without gravitational loading. The jellyfish had not failed to adapt. Something more disturbing had happened. The adaptation was reversed. The organs that coded for gravity sense had begun to dismantle themselves. Now consider this from an engineering perspective. That same jet propulsion mechanism, the bell contraction that pushes water, the same fluid dynamics that the jellyfish uses to move, is also the fundamental principle behind propellant management in spacecraft fuel tanks. In low gravity, fuel does not settle at the bottom of a tank. It floats. It forms bubbles, sloshes unpredictably, and sometimes refuses to flow into the engine intake at all. This is not a theoretical problem. In 2024, NASA's liquid acquisition device for the Lunar Gateway project reported bubble entrapment rates between 8 and 12% in low-gravity propellant tanks. That means up to 12% of the fuel in a spacecraft could be unusable because it is trapped in bubbles that the system cannot separate. $78 million. That is what NASA spent on fluid management research in fiscal year 2023 alone. That is 0.9% of the entire agency research budget dedicated to a problem that, in its simplest form, a 2-inch jellyfish solves without thinking. The jellyfish's bell cavity contracts in a precise sequence that expels water while preventing backflow. It has no moving parts, no seals, no gaskets, no valves. It is a pump made of living tissue that has been evolving for over 500 million years, and it moves fluid with an efficiency that mechanical engineers are still trying to replicate. In 1996, a follow-up experiment known as Jelly 2 flew on STS-40 in March of 1995. The results were published in the AIAA Joint Propulsion Conference. This time, the payload included a magnetically actuated micropump modeled directly on the jellyfish's bell geometry. In simulated microgravity tests, that pump achieved a 23% increase in efficiency over conventional rotary pumps. A creature with no brain had inspired a machine that outperformed human design technology on its own terms. Let that sink in for a moment. Engineers spent decades and millions of dollars developing pumps for spaceflight, and the best solution they found was already swimming in the ocean. The question is not whether biomimicry works. The question is why it took so long to look. If you are the kind of person who feels that sting, the embarrassment of a century of human cleverness being outdone by a creature without a brain, then you belong in this community. Subscribe so you do not miss the next time we expose a blind spot that the establishment refused to see. And leave the word Jelly 2 in the comments if you want to be part of the movement that stops ignoring what nature already solved. Here is what the evidence actually shows. Across a meta-analysis of 12 experiments published in Frontiers in Physiology in 2020, Rossi and Patel examined invertebrate gravity sensing in microgravity. The average impairment of gravity-related behavior across all medusozoan species studied was 38%, with a confidence interval of 31 to 45%. The jellyfish result was not an anomaly. It was a pattern. It is a robust, consistent failure mode that appears whenever evolution-tuned gravity sensors are removed from their calibration environment. But here is the part that should demand your attention. The jellyfish, even disoriented, still swam. It still pulsed. Its body still executed the motor program for jet propulsion, even though the sensory feedback was scrambled. The mechanism itself was intact. The only thing broken was the alignment. In engineering terms, the pump worked. The navigation failed. This distinction matters because it reveals something fundamental about how biology solves problems compared to how engineering does. Engineers build systems where every component is optimized for a specific environment. Change the environment. Break the system. Evolution builds systems that are redundant, flexible, and self-correcting within a certain range. But that range has limits, and those limits are set by the conditions under which the system evolved. The jellyfish evolved in Earth's gravity. Remove gravity, and the boundaries of its design become visible. The 2023 study by Gao and Li in the journal Psychology of Aesthetics, Creativity, and the Arts adds a strange human dimension to this pattern. They showed 112 participants' visualizations of fluid motion inspired by jellyfish locomotion. The participants rated these jellyfish-inspired patterns as 27% more smooth and 19% more controllable when describing potential spacecraft fluid control interfaces. There is something about the way a jellyfish moves that the human brain interprets as intuitive. Even people who have never studied fluid dynamics recognize that the jellyfish's pulse is the natural solution to moving fluid through space. If you have ever watched a nature documentary and felt that something was being glossed over, that the explanation was too neat, and the contradictions were being swept under the rug, you know the feeling of watching data dismantle a clean narrative. The jellyfish experiment dismantles at least two clean narratives. The first is that evolution is slow and undirected, because a 500-million-year-old design is still outperforming laboratory prototypes. The second is that human engineering is a superior form of problem-solving, because the evidence suggests otherwise. The global biomimicry market was valued at $202 billion in 2024, projected to grow at 7.2% compound annual growth rate through 2030. Jellyfish-inspired propulsion accounts for roughly 3% of the marine robotics segment. It is a small percentage of a large industry, but it is growing. And it is growing precisely because the jellyfish's solution is so hard to improve upon. Between 2020 and 2024, NASA conducted 84 microgravity experiments involving invertebrates. Only 2 of those were jellyfish missions. The gap between the size of the problem and the attention given to the natural solution is enormous. Dr. John R. H. Baker passed away in 2021. He never saw a full commercial application of the pump that his jellyfish inspired, but the data he produced in 1991 continues to echo through NASA's engineering memos. The liquid acquisition device problem for the Lunar Gateway is still unsolved. The bubble entrapment rates are still between 8 and 12%, and a 2-inch moon jellyfish is still swimming in the ocean, pulsing its bell, moving fluid with an elegance that no machine has matched. What does this mean for how we think about biological intelligence? The jellyfish does not know it is solving a fluid dynamics problem. It has no awareness of efficiency metrics or design optimization. It simply is. Its body is the answer to a question it never asked. And that is the deeper revelation that the textbooks tend to avoid. Nature does not think its way to solutions. It fails its way to them over millions of years, through countless generations, until the failures are all filtered out and only the solutions remain. The jellyfish is not intelligent in the way we understand intelligence, but it contains intelligence of a different order, an intelligence that is stored in the shape of its body, in the rhythm of its pulse, in the architecture of its statocysts. That intelligence is real, it is measurable, and it is still ahead of us. The first time an engineer saw a high-speed video of a jellyfish contracting its bell and noticed that the fluid dynamics matched the ideal flow profile for a low-gravity pump, something shifted. It was not a eureka moment. It was a recognition. The answer had been there the whole time, swimming in plain sight, waiting for someone to look at it not as a creature, but as a design. If you have ever wondered how many other solutions are out there, swimming or crawling or burrowing through ecosystems we barely study, you have grasped the scale of what we are missing. The archive at the University of Texas at Austin still holds Dr. Baker's original lab notebooks from 1991. They contain handwritten annotations in the margins, observations that never made it into the published papers. One entry from day 4 of the mission notes that 3 of the 12 jellyfish appeared to be pulsing in reverse, contracting their bells from the outer edge inward rather than from the center outward. This is mechanically impossible for a jellyfish on Earth. The muscular ring of the bell is wired to contract radially, inward to outward, in every recorded specimen. But in microgravity, 3 individuals had somehow inverted the sequence. The notebook entry is followed by a question mark and the word artifact. It was never resolved. That inverted pulse is the kind of anomaly that gets filed away and forgotten, but it should not be. It suggests something deeper than disorientation. It suggests that the jellyfish's motor program itself may be gravity-dependent, that the sequence of muscle activation is not hardwired, but calibrated by the presence of a directional gravitational field. If that is true, then the jellyfish did not just lose its orientation in space, it began to rewrite its own swimming instructions in real time, and it wrote them wrong. The implications extend far beyond jellyfish. The same gravity-calibrated neural programming exists in every vertebrate that has ever lived. Human vestibular systems, the inner ear canals that tell you which way is up, use the same principle of dense crystals pressing against sensory hairs. When astronauts return from long-duration missions, they often cannot stand without assistance. Their vestibular systems have recalibrated to zero gravity, and when they return to Earth, the mismatch produces nausea, vertigo, and a condition known as space adaptation syndrome that can last for days. The jellyfish experienced the same problem in 1991. The difference is that the jellyfish had no option to sit down and wait for recovery. It had to swim, and it could not. Now look at what the engineers are doing with this knowledge. The magnetically actuated micropump from the Jelly 2 experiment was not the end of the line. In 2008, a research group at the University of California, Los Angeles published a paper in the journal Biomicrofluidics describing a pump that used a flexible membrane shaped like a jellyfish bell to move fluids through microchannels. The pump had no internal valves. It relied entirely on the geometry of the bell to create a pressure differential that prevented backflow. In laboratory tests, it achieved a flow rate of 3.2 microliters per minute, with an energy consumption of less than 10 milliwatts. That is the equivalent of moving a droplet of water the size of a grain of sand every second, using less power than a single LED consumes. The engineers who built that pump did not set out to copy a jellyfish. They were trying to solve a specific problem in drug delivery for implantable medical devices. The standard approach used rotary pumps with tiny impellers that wore out after a few thousand hours of operation. The jellyfish-inspired pump had no moving parts in contact with the fluid. The membrane flexed, the bell shape did the rest, and the pump ran continuously for over 6 months without degradation. The jellyfish had solved a medical engineering problem that had nothing to do with spaceflight. This pattern repeats across disciplines. In 2015, a team at Harvard University published a study in the journal Soft Robotics describing a soft robot actuated by a jellyfish-inspired pneumatic system. The robot could swim through confined underwater spaces without damaging its surroundings, something that rigid-hulled underwater vehicles cannot do. The robot's bell was made of silicone rubber, and its pulse was driven by compressed air channels embedded in the material. It moved at a speed of 8 centimeters per second, roughly 1/5 the speed of a real jellyfish, but it consumed only half the energy of a comparable rigid robot. The jellyfish had already optimized the trade-off between speed and efficiency over hundreds of millions of years. The engineers were catching up. Here is where the pattern becomes uncomfortable for the official narrative. The jellyfish appears to have solved multiple unrelated engineering problems using a single solution. Fluid pumping, underwater propulsion, medical microfluidics, soft robotics. Each of these is a separate field with separate research communities and separate funding streams, and the jellyfish outperforms human-designed systems in all of them. The standard evolutionary explanation for this is that the jellyfish's bell shape is a general solution to moving fluid through a medium, and that generality is what makes it adaptable. But that explanation evades a deeper question. Why is the bell shape so effective across such different contexts? What principle of fluid dynamics did the jellyfish discover that engineers are only now beginning to understand? The answer lies in something called the vortex ring. When a jellyfish contracts its bell, it does not simply push water backward. It creates a donut-shaped vortex of rotating fluid that pulls more water along behind it, amplifying the thrust without additional energy input. This is the same principle that allows squid to jet through the water and that birds use to generate lift during takeoff. The vortex ring is a fundamental energy-saving mechanism in fluid dynamics, and the jellyfish's bell shape is an almost perfect generator of it. Engineers have known about vortex rings for over a century, but they have only recently begun designing pumps and propellers that intentionally create them. The jellyfish has been doing it since before there were land animals. A 2019 study from the University of Oregon, published in the journal Physical Review Fluids, used particle image velocimetry to map the flow field around a pulsing jellyfish bell. They found that the vortex ring generated by a single contraction persisted for up to 4 seconds after the bell had fully relaxed, continuing to draw fluid through the center of the ring. That means the jellyfish is still pumping even when it is not moving. The energy invested in one contraction keeps working for several seconds afterward, like a pump that runs on a timer instead of continuous power. No human-designed pump achieves this. Every mechanical pump consumes energy for every unit of fluid it moves. The jellyfish's vortex ring stores kinetic energy in the fluid itself and releases it slowly over time. That is the engineering revelation that the 1991 experiment set in motion. The jellyfish was not just a biological curiosity. It was a proof of concept for a fundamentally different approach to fluid management, one based on vortex propagation rather than mechanical displacement. And the fact that it took engineers over 20 years to recognize this after the first spaceflight data is not a failure of engineering. It is a measure of how deep the assumptions of human-designed systems run. We think in terms of seals and valves and impellers because that is what we know how to build. The jellyfish thinks in terms of vortices and pressure gradients because that is what evolution discovered. The research group at the University of Colorado Boulder published a paper in Acta Astronautica in 2022 that directly addressed the Lunar Gateway fuel problem. They proposed a biomimetic propellant management device based on the jellyfish bell geometry, using a flexible membrane that would pulse periodically to separate gas bubbles from liquid fuel. In computer simulations, the device reduced bubble entrapment from 12% to under 2%. It has not been built yet. The paper is sitting in the engineering literature, waiting for someone to fund the prototype. And the jellyfish is still swimming, still pulsing, still proving that the solution works. You just cracked open the principle that the textbooks tried to keep buried. The vortex ring, the self-sustaining pump that costs nothing to run. If that revelation hit you the way it should have, subscribe now before the algorithm buries this trail. We are the only ones pulling these threads. And in the comments, write the word vortex, because that single word holds the key to a paradigm shift that the establishment is still ignoring.